Hi friends. Welcome to a new learning video on PharmaGuideline. Today we will discuss sampling in cleaning validation. Cleaning validation plays a crucial role in the pharmaceutical industry to ensure that equipment, surfaces, and utensils used in manufacturing are effectively clean, preventing cross-contamination and maintaining product quality. A key component of the cleaning validation process is sampling, which involves the collection and analysis of samples to assess the level of residues or contaminants present. First of all, let us know the importance of sampling in cleaning validation. Sampling is essential in cleaning validation as it provides a representative assessment of the cleanliness of equipment and surfaces. It helps to identify any potential residues or contaminants that may remain after the cleaning process. By analyzing samples, pharmaceutical companies can evaluate the effectiveness of their cleaning procedures, identify areas for improvement, and ensure compliance with regulatory requirements. Now, let us know the key considerations for an effective sampling strategy. Sampling locations. Selecting appropriate sampling locations is crucial to obtain representative samples. These locations should include critical areas that are prone to contamination, such as equipment surfaces, joints, crevices, and areas that are difficult to clean. Consider worst-case scenarios and areas with the highest risk of residual contamination. Sampling techniques. Different sampling techniques can be employed, including swabbing, rinsing, direct surface sampling, or even equipment disassembly in some cases. Each technique has its advantages and limitations, and the choice of technique depends on factors such as the nature of the equipment, surface, or residue being sampled. Sample collection. Ensure proper collection techniques to minimize the risk of cross-contamination or sample degradation. Use clean and validated sampling materials, follow appropriate procedures for sample collection, and prevent exposure of samples to external contaminants during collection and transportation. Sample analysis. Analytical methods should be selected based on the nature of the residues or contaminants to be detected. Methods such as chromatography, like HPLC, GC, spectrophotometry, or specific tests for microbial contamination can be employed. Ensure that the selected methods are validated, accurate, and reliable. Sample size and frequency. Determine appropriate sample size and frequency based on statistical considerations, regulatory requirements, and the level of confidence required. Adequate sample size and frequency increase the likelihood of detecting potential residues and provide a more comprehensive assessment of the cleaning process. Documentation and record keeping. Proper documentation of the sampling strategy, including sampling locations, techniques, analysis methods, and results, is crucial for traceability and audit purposes. Maintain detailed records to demonstrate compliance with cleaning validation requirements. Sampling is a critical aspect of cleaning validation in the pharmaceutical industry. It allows for the assessment of residual contaminants, ensuring effective contamination control. By implementing a well-designed sampling strategy that considers key factors such as sampling locations, techniques, analysis methods, and documentation, pharmaceutical companies can enhance the reliability and accuracy of their cleaning validation process. This, in turn, promotes product quality, patient safety, and compliance with regulatory standards. Like this video, and share it with your friends and colleagues. Don't forget to subscribe to this channel and also hit the bell icon to get regular updates. Thanks for watching this video.